Okay, so students, today's class is mainly concerned with the basic aspects of pesticide residues. We won't be going into pesticide residue analysis as such. We will learn something about the residues, what is its definition, what are the different kinds of residues, and what is the different terminologies which we use and how we arrive at a MRL and all, and what is the concept behind MRL, what is the concept behind uh, this one, ADI, reference doses, etc. So that is the thing which we will learn today. First we will go into the pesticide residue analysis part and the definition of that. Are you able to see the slides now? Okay, fine. So first we will go into the definition. Okay. I have given two definitions. Okay. I have given two definitions, of which the second FAO definition is the one which I usually prefer, though basically both the definitions doesn't vary much in their meaning. Residues is defined by Food and Agriculture Organization as substance or substances, because it can constitute residue from a single pesticide or from two pesticides, or sometimes what can happen is that if you spray a pesticide, it can lead to the formation of another compound which is also as toxic as the parent compound. So we have to measure both the things. I will take the example we discussed about pro insecticide. Malathion is toxic, its degradation product malloxone is also toxic. So here, if you want to calculate the residue from malathion, you have to sum up the residues of both malathion as well as malloxone. I will repeat. If you are analyzing the residues of malathion, if you are spraying malathion, if you are analyzing malathion, you have learned that malathion is a pro-insecticide, and once it is sprayed in the crop, it gets converted into a more toxic malloxone. So malloxone is another compound which is formed because of the use of malathion. So if you are using malathion, you have to analyze malathion residues as well as malloxone residues, and sum the residues of malathion and malloxone. Then only you have to depict the value of residue from malathion, because malloxone is another toxic compound. That presence of malloxone is due to the use of malathion. So it can be a single substance or a substances. Sometimes we may use pesticide mixtures. In that case, we have to measure both the pesticides. Chlorpyrifos, pesticide mixture if you are using, then we will get residues of both chlorpyrifos as well as cypermethrin. So substance or substances which remain in or on. Here it's in or on. On means if you are using a contact pesticide, it will be present outside the commodity. If you are using a systemic pesticide, that pesticide will be remaining inside the commodity. So that terminology you can differentiate inside the commodity or sometimes outside the commodity. So residues can be a substance or substances which which is remaining in or on food. Food means anything which is consumed by human beings. Feed includes anything which we are feeding to animals. It can be cattle feed, it can be poultry feed, because whatever be the feed which are providing to livestock, it is getting into the food chain and ultimately it will reach human beings. So we have to measure residues in food commodity which human beings consume, or feed which is provided to animals, domestic animals, And we sometimes measure the residues in environment. It can be in soil, it can be in air, it can be in water, following the use of a pesticide. That is critical. Once we use a pesticide, we check for the presence of a substance or a substance which is remaining in or on a food commodity, a feed commodity, or in the soil, air, or water. Invariably, it must be after the use of a pesticide. So, this is the basic definition of pesticide residues. And one more thing here that we have discussed earlier, substance or substances, this definition includes one more thing. For regulatory purposes, it includes the parent compound. That is, we have taken the example of malathion and any specified derivatives such as degradation and conversion products, metabolites, and impurities. Degradation, conversion products, metabolites. This include malathion to malloxone, DDT to DDE. Both DDT as well as DDE is toxic. DDE is a degradation product. Malathion to malloxone is a conversion product or a metabolite. So, whatever be the parent compound, we have to measure it, its degradation, metabolite, or conversion product, or sometimes some pesticide formulation may have some impurities which have toxicological significance. So, the residue should include the parent compound, that is the pesticide, along with its metabolites, degradation or conversion product, or any impurity. And that degradation, conversion product, or metabolite or impurity should have toxicological significance. See, sometimes what happens is that malathion to malloxone, after conversion to malloxone, this malloxone will get again degraded into a diol form. Two hydroxyl group will be added to this malloxone. Once this hydroxyl group is added, it becomes non-toxic. So we need not measure this hydroxyl form of malathion. We have to measure only those products or the parent compound which is toxic to human beings or animals. So residue is by definition of FAO, it is a substance or substances which remain in or on food, feed, soil, air, or water after the use of a pesticide. And we should take into consideration the parent compound, its metabolites, its degradation products, its conversion products, and any impurity which is toxic to living organisms. Are you clear regarding the definition? Yes or no? Hmm? No answers? Yes. Okay. Abhishek? Abhishek, are you there? Oh, okay, fine. So, if you see the WHO definition, it's almost remain the same, but meaning is almost the same. Here, substance or a mixture of substances, the same thing in food for man or animals. That includes food, feed, resulting from the use of a pesticide. Here, one thing is that here the environment part is not considered, soil, air, or water, but that is critical in the case of pesticide residue analysis. So, food or feed resulting from the use of a pesticide, and it includes pesticides, its derivatives such as degradation, conversion products, metabolites, reaction products, and impurities that are of toxicological significance. So, by meaning, it's same, but FAO, I prefer FAO because it is more inclusive in nature and it's more easy to understand. And coming to the next definition, that is pesticide residue analysis. It is defined as the qualitative and quantitative analysis. Qualitative means we can say that whether a sample is having pesticide residue or not, whether a sample is contaminated or not. Quantitative means we can determine how much pesticide is present. So we can pesticide residue analysis defined either as a qualitative or a quantitative step of representative samples. The sampling is in pesticide residue analysis is critical. We will move into sampling once we go into the steps of pesticide residue analysis. Today we won't be going into that. We will be analyzing representative samples. Sampling should be representative, otherwise all the data will go wrong. So samples should be drawn perfectly, and we usually draw samples from agriculture fields from the farmer itself. We usually call it as farm gate samples. Repeat the term farm gate sample, because we usually sample from the field itself, or we usually sample it from markets. We call it as market sample, or we can sample it from the environment. Environment means soil, air, or water. That is from environment, and we check for pesticides or its toxic metabolites. So pesticide residue analysis is defined as qualitative or quantitative analysis of representative samples, and these samples which we draw from market, environment, or field or farm gate samples, and we check for the presence or absence of pesticides and its toxic metabolites. So I think it is the both definition, residue definition and residue analysis definition is now familiar to you. Now we will go into the types of residues. How residues are classified and what are the different kinds of residue. The first kind of residue is surface residue or effective residue. So what we will do is consider a scenario. If someone has sprayed a pesticide in a crop, I think you might have sprayed at least one or the other pesticide. Even a biocontrol agent you might have sprayed in a crop. If you spray a pesticide in a crop, if you spray it over a crop, if the spray is a bit excess, it will start dripping out off from the leaf surface. And after dripping of the leaf surface, dripping of the excess spray fluid, there will be some quantity of pesticide remaining on the treated surface, treated leaf or plant surface. So the pesticide residue remaining after the initial deposit. We have sprayed a quantity, but some quantity of pesticide has fallen off the leaf area or crop surface because we have sprayed a bit excess of the spray fluid, and whatever the portion of the pesticide remaining on the crop surface is called as surface residue or a effective residue. We might have sprayed say 1 liter of pesticide onto the crop, but the effective residue will be only a part of it. Some part of the residue must have fallen over to the soil. So surface of residue or effective residue is the portion of pesticides left over from the initial deposit. So that is surface or effective residue. So that is immediately after the spray, after the excess pesticides residues are dripped off, then whatever else is remaining, that is surface or effective residue. Coming to the next category, that is called as dislodgeable residue. Okay, you have sprayed the field. After some time, the excess pesticide has fallen off the crop surface, and surface or effective residue is remaining. And we enter into the field again. And the pesticide might not have entered into the crop tissues. The pesticide must not have properly adhered to the crop tissue. If you enter the field immediately after the spray of a particular pesticide, you are at greater risk, because once you are entering into the field immediately after the spray, your body will be coming into contact with the sprayed surface, and you will be removing a part of the pesticide residue, and that portion of the pesticide residue which is easily removable from a treated surface is called as dislodgeable residue. I will repeat again. If you are using a pesticide in a particular crop, say rice, and after the spray you are entering the field again. So once you enter the field again after the pesticide spray is completed, some part of the sprayed pesticide might get dislodged from the crop surface and may cause harm to the person who is entering into the field. You can imagine if you're entering into rice field, your body will be coming into the contact with the leaf tissue on which the pesticide was sprayed a half an hour or 1 hour before, and since it was not absorbed into the plant tissue, it will be easily removable and your body will get contaminated. So that portion of residue which is easily removable from a treated surface is called as dislodgeable residue, and this is the portion of residue which is which poses a risk to farm workers, because these are the persons which get easily contaminated with dislodgeable residue. If you are moving through any Munnar or any area, you might have seen during a tour and all, tour program and all, in Kannandevan tea plantations and all, keep a board in the tea plantations after the pesticide spray. This is a pesticide sprayed area, don't enter. Have you seen, anyone has seen that? Especially in bigger plantations and all, you might have seen. Yes, that is to prevent risk to farm workers and other persons who unintentionally get inside a sprayed area and might get exposed to dislodgeable residue. So that is dislodgeable residue. And the next term is penetrated residue. We have surface residue, that is the residue remaining in the crop surface after the initial deposit. Just a minute. Sorry. എപ്പോഴാ? ഓക്കെ ഓക്കെ, കുഴപ്പമില്ല സാർ. ദീപ്തിയുടെ ഞാൻ നോട്ടെ, ദീപ്തിയുടെ ഫയൽസ് ഒന്ന് എടുത്തിട്ടുണ്ട്. So penetrated residue after the surface, after we spray pesticide, the surface residue will be remaining, and the residue remaining into this on the plant surface will get penetrated into the plant tissue. That is penetrated tissue, especially in case of systemic insecticides, translaminar pesticides, etc. It will penetrate into the plant tissue. So that becomes penetrated residue. The surface residue becomes penetrated residue by migrating into the substrata. So that is penetrated residue. Then comes terminal residue. These terminal residues are nothing but breakdown products of pesticides which are stable and create as many problems as original compounds. That means if you mainly with respect to persistent pesticides such as DDT, BHC, etc. DDT and BHC will get broken down into their respective metabolites, and certain metabolites of this DDT organochlorines, DDT and BHC are as stable and as toxic as the parent compound, that is DDT or BHC. So it may get degraded, the parent compound may get degraded, but the breakdown products are as stable and is as toxic as the original compound. So those residues are called as terminal residue. We usually don't get terminal residues in the case of new pesticide molecules, but in the case of organochlorines and all, we may get some metabolites which are stable and toxic. And coming to the next residue, it is conjugate residue. You studied about glutathione as transferase. And what does the reaction of what is the reaction of glutathione as transferase? Do you remember that? Studied it in pesticide insecticide resistance, mechanisms of insecticide resistance. Radical, no. It catalyzes conjugation reactions. Glutathione as transferase catalyzes conjugation reactions. It catalyzes conjugation of pesticides with sugars, carbohydrates, etc. So it catalyzes conjugation reactions. So the pesticide residue undergoes secondary metabolism. That metabolism can be with glutathione as transferase, and the pesticides or its metabolites forms conjugates with sugars, amino acids, etc. And these conjugates can pose a toxic conjugate to the human beings or other organism. That means the conjugation product of pesticide or its metabolites with sugars, amino acids, or any substrate from the plant body can be as toxic as the pesticide molecule. So that is conjugate residue. The product of conjugation reaction that lead to the formation of a conjugation product, a residue which is toxic to other organisms. And then the next term is bound residue. Bound residue is nothing but pesticides or its metabolites which it's are not easily extracted by the usual extraction method. Whenever we are going for pesticide residue analysis, we use a step called as extraction. Extraction is nothing but extracting the residue from the substrate. It can be from it can be from fruits, vegetables, spices, cereals, etc. So we use different kind of methods to extract the pesticide, but there are certain pesticides which are not easily extracted from the substrate, and these residues are called as bound residues, which are not easily extracted from the commodity. Coming to the next term, that is aged residue. Aged residue is nothing but, I will give you an example. We had certain reports that the endosulfan was last spray of endosulfan was carried out in Kasargod. If I remember it correctly, it was in 97 or 98. And in 2003 or 04, there were certain studies from by certain groups, and they reported that endosulfan residues are still remaining in water, soil, and even in the blood of persons residing in Kasargod area. So if that reports are true, there are certain scientists who argue against that reports. They say that scientifically that reports are wrong. So if hypothetically, if we say that that report is correct, that means even after we are stopping the pesticide application for 4 or 5 years, still the residue is remaining, then that residue is called as aged residue. By definition, I will say the pesticides and its metabolites present in the environment after an application and following a period long enough to allow transportation, metabolism, and dissipation process to alter the distribution and chemical nature of some of the applied residue. That means in 98, Plantation Corporation applied endosulfan, and in 2003 and 4, after about 5 to 6 years, again certain people are claiming that they have found residues of endosulfan as well as endosulfan sulfate, that is another metabolite of endosulfan, and that is after a long period of time, after 4 to 5 years. And those kind of residue can be termed as aged residue. So, are you clear with respect to these definitions of residue? Surface residue, penetrated residue, dislodgeable residue, bound residue, conjugate residue, terminal residue, age residue. Yes, okay, okay, fine. Then we'll come into the objectives of pesticide residue analysis. For what purpose we are going for pesticide residue analysis? What are the objectives of pesticide residue analysis? The first objective is the kinetics of pesticide metabolism. That means in pesticide residue analysis we go for the kinetics of pesticide. Usually agriculture chemicals people do it. Kinetics of pesticide metabolism, they will study it in water, they will study it in soil, they will study it in plant system, because in every environment the kinetics varies. I will take the example, say in crop. So what they will do is, today we are spraying the pesticide. Today itself they will take a sample. First day they will take a sample. That means tomorrow. Third day they will take a sample. For fifth day they will take a sample. Seventh day, ninth day. Like that repeatedly they will be taking samples and they will be analyzing the residue. So usually it should follow a declining trend. So today, if you are taking, we call it a 0th day sampling. If you are spraying today and if you are sampling it today itself, we call it a 0th day. Next day is called as first day. So for immediately after spray, there will be residue will be more. That is usually that residue we can call it as surface residue. Then after on first day we'll be taking residue. Usually it should be less than the 0th day residue. Third day it should decrease. But these dissipation pattern, we call it as dissipation pattern. These pattern vary with pesticides. Certain pesticides are more persistent. So that means the graph will not be a steep declining slope. It will be, the slope will not be that steep. It will be following a small gradual reduction of the pesticide concentration. But if it is if pesticide is non-persistent, it will decline pretty sharply. So these kinetics can be understood by pesticide residue analysis. And next, pesticide residue analysis will help us to determine MRL. I think you know the full form of MRL. Do you know? Anyone? Maximum residue limit. Yes, okay. Then, based on pesticide residue analysis, we establish waiting period. I will discuss how MRL is determined and how waiting period is established. So I'm not touching it over here, because that is one thing which we will study today itself after this slide. So it will help us to determine the maximum residue limit, help us to establish waiting period, and helps us to determine the pesticide use pattern by farmers. Here in our pesticide residue lab, we are analyzing the residues mainly from 4 districts: Ernakulam, Palakkad, Thrissur, and Malappuram. And there is a clear trend in the pesticide use pattern in this district. We usually get chlorpyrifos residues from Ernakulam district. We don't get usually chlorpyrifos residues from fruits and vegetables in Thrissur, Palakkad, and Malappuram district. But we get chlorpyrifos residues from Ernakulam district. One reason might be in Ernakulam, especially in the Ernakulam, the major vegetable growing tract is in the eastern part, that is Perumbavoor, Muatuvuzha, Kodamangalam, that belt. In that belt is famous for pineapple cultivation also. So chlorpyrifos is one pesticide which is commonly used in among pineapple farmers, and This pesticide is commonly available in the pesticide dealers also. So whenever a farmer goes to a pesticide dealer, he will readily give a pesticide containing chlorpyrifos. Because in my locality also, I know whenever a farmer goes to the pesticide local pesticide dealer, that pesticide dealer it's near to my home. He usually gives a pesticide Hamla. Hamla is trade name is Hamla. I think that is chlorpyrifos plus cypermethrin. Otherwise, he will give chlorpyrifos, Hilban. That is the trade name. So it varies. So if you want to find out the pesticide use pattern of farmers in that area, you can usually determine by collect the samples from that area, analyze it, and you can know which all pesticides are used by the farmers in that particular area. So we'll get the pesticide use pattern. And next, we can conduct risk assessment based on dietary intake of pesticide. That we will detect. MRL waiting period dietary intake that we will discuss in the next slide. And we can study the effect of storage and processing methods on persistence of pesticide risk. That is one thing. I will ask you one question. You have residue in a fresh commodity, and what will be the concentration of residue after cooking, whether it will increase or decrease? Why no answer? I will repeat the question. You have a fresh vegetable, you are cooking it. Say you have bindi, you are preparing a sabji with bindi or sabji or any food item dish with bindi, and what will be the residue in a cooked product? Abhishek has given it may be decrease. What about Shika and Rahul? Increase? Don't know. Usually it decreases, especially with Indian mode of cooking, because we usually deep fry our vegetables. We overcook the vegetables. We usually say that the Indian method of cooking is one of the most destructive method of cooking. We usually destroy all the antioxidants present in a particular vegetable or fruit because of our cooking process. If you see Oriental dishes or Western dishes, they usually half cook the items. They don't even, mostly they prefer to eat as a salad. So it preserve the natural nutrition of that particular commodity. Take our case, our Indian, if you are taking any vegetable, say bindi, brinjal, or say any bitter gourd, snake gourd, what we usually do is in Kerala itself, I don't know in uh, Karnataka, Abhishek, Kerala, what we will do is we will first boil it, and then we will add oil. We'll heat oil and then deep fry, sometimes stir fry those items. അല്ലേ അങ്ങനെയല്ലേ നമ്മൾ എണ്ണയിലിട്ട് വറുത്തെടുക്കുകയാണ് ചെയ്യുക മോസ്റ്റ്ലി. We will deep fry our things in oil. And see, the temperature of oil is far higher than any other thing. So if you are cooking in such high temperature, usually residues will dissipate. Any cooking process will reduce the pesticide residue. I will give you a few papers. I will attach it. You can go through it, wherein there are several reports which say that cooking reduces pesticide residues drastically. And depending upon the cooking method, it varies. Deep frying varies. If you are preparing curry, say sambar or any other thing, then the decrease in vegetable residues is in another manner. So it depends upon the cooking method also affects the decrease in pesticide residue. But normally cooking will all reduce the pesticide residue. If you want to analyze how storage or processing methods affect the pesticide residue, we have to go for pesticide residue analysis. Then we have to monitor food and food commodities for pesticide residue. That is what we do over here. We collect the samples from markets of farmers and we check for the presence or absence of residues, and that we represent it based on FSSAI Act, Food Safety and Standards Authority Act. We say whether it is above the FSSAI MRL or below MRL or not. And in certain labs, we usually don't do it because we are not accredited lab. Accredited labs can analyze pesticide residues for global trade. That means an accredited lab, if they give a certificate that this commodity is free of pesticide residue, it is accepted all over the world. So if that certificate has to be valid, it should be from a accredited lab. Our lab is not accredited, so our lab is not considered for the certificate from our lab is not considered for global trade. Our certificate usually we give it based on FSSAI Act. So the major purpose of carrying out pesticide residue analysis is to study the kinetics, to determine the MRL, to determine the waiting period, to check the pesticide use pattern of farmers in a particular area, and risk assessment based on dietary intake. MRL, waiting period, and dietary intake we'll discuss later. Then how processing and storage affect the pesticide residue, that can be determined using pesticide residue analysis. Monitoring of commodities for national trade as well as global trade. So these are the objectives of pesticide residue analysis. Okay. Now we will come into the different terminology. Uh, Please try to understand this terminology very well. If you're not clear with any terminology, you can raise your, you can unmute your mic and ask a questions whenever you are having any doubt. Because after this, after one or two definitions, I will ask you to unmute your mic and write up a small calculation in your so that you have to tell something. Okay. The first is no observed adverse effect level, NOAEL. Okay. So consider Abhishek has discovered a new pesticide molecule and Abhishek wants to check its toxicity before carrying out before when once you have discovered a new pesticide molecule, first thing you have to do is go for its toxicological assay, whether it is toxic to other organisms. You might have screened it against insects and you might have found that yes, it is very effective against insects, so you are satisfied with this, with its insecticidal activity, but you have to check its toxicological significance, whether it is toxic to other organisms. So what you will do is you will take some lab animals. You can all three of you can unmute your mic, and what are the common lab animals in which we can carry out toxicological studies? Any,
White mice.
huh?
Mice.
Mice. Okay.
White mouse.
Okay. What albino rat,
Rabbits.
mice, rabbit. So
Rabbits.
for rabbits, yes. So
Pigs.
Abhishek
Pigs.
took some rabbits. He took 10 rabbits. He gave each rabbit different doses of this pesticide molecule, what we have discovered, daily over its entire lifetime. I will repeat what I have said. Abhishek took 10 rabbits, and for each rabbit, he will give a particular dose of the pesticide molecule which he has discovered along with food throughout its lifetime. Okay. So the rabbit number one will receive a particular dose. Rabbit number two will receive a particular dose, a dose higher than the one given to the rabbit number one. Rabbit number three will receive a bit higher dose again. Four will receive another higher dose. So you have a set of animals which receive a particular dose of that particular chemical every day through its lifetime. Every day, remember that. Every day he will mix, he will along with the food which is provided to the rabbit, Abhishek will mix that particular pesticide, particular quantity of pesticide, and give that to that particular animal. And he observe all these 10 animals carefully, and if something happens to a particular animal, he will record it. By the time when the animal gets died, it can die off because of the poisoning from the insecticide, or sometimes the animal will get killed because of natural age related ailments. So after some time he will conduct a postmortem of the animals, and he will check whether the pesticide has caused any abnormality to that particular organism. We usually say that the pesticides can harm our internal organs, can harm our kidney, can harm our heart, can harm our lungs, it can cause tumors. So after the death of the animal, he conducts a postmortem and check for any of these ailments in that particular animal. And he among the five, among the 10 animals, he found that Abhishek found that from the sixth animal onwards, he was increasing the dose from 1 to 10. From each animal he was increasing the dose. From the sixth animal onwards, there was some abnormality in the internal organs, but the fifth rabbit, which up to fifth rabbit, 1 to fifth rabbit which received the chemical, didn't show any adverse effect in the internal organs or the animal. So even though these five animals were fed with pesticides daily over its entire lifetime, it was not showing any any adverse impact on that organism. So the fifth animal which received that particular dose, it just received a higher dose compared to the first, second, third, and fourth animal. So the dose which didn't cause any adverse effect to that organism, even though it was fed with that particular pesticide daily throughout its entire lifetime, that dose is called as no observed adverse effect level, NOAEL. Are you clear? Yes or no? Yes. Yes. What about Rahul? Yes. Okay. So here, one thing you should remember is it is the maximum concentration. I told you Abhishek took 10 rabbits, and with each rabbit 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, he has increased the dose for each animal. And up to the fifth animal, there was no adverse effect, even though these five animals were fed with that particular chemical. Only the sixth animal showed, from sixth animal onwards, we had some adverse effect. So we take the dose which was given to the fifth animal. That was the maximum dose among these five animals, 1 to 5. So that maximum concentration which we have provided in a diet to that particular animal, which has not caused any adverse effect, that is called as no observed adverse effect level. That is, the unit is milligram per kg body weight per day. That means the quantity of the pesticide, we express it in milligram, and body weight of the animal will be in kg. And every day we are providing the pesticide. So that's why the unit is milligram per kg body weight per day. Okay. So I will ask you another question. Can you say that this value you have got, Abhishek has got one value, say, is saying that 1 milligram per kg body weight per day. If you consume, rabbit is consuming daily, it doesn't cause any harm. Above that, it can cause some damage to the animal. So 1 milligram is safe for the rabbit, and this 1 milligram per kg body weight per day will remain safe for an human beings also. Whether that statement is correct or not, is saying that 1 milligram per kg body weight per day, if a rabbit is consuming, it is not causing any adverse effect. But so it is this same concentration, 1 milligram per kg body weight per day, will be safe for human beings. False. Why? Abhishek, you can unmute and tell why.
Sir, mechanism and body weight will vary, sir, compared to animals.
Body weight, that factor, if you
Yes,
that is
yes,
per
sir.
kg body weight, that is
But
no.
mechanism of resistance.
Yeah, every organism is different. So if
Ah,
we have got a data from one organism, we can't apply it to another organism. That's why we call LD50 for rats, LD50 for rabbits. If you see LD50 for different LD50 for different pesticides, different organisms, you get different values. So what we will do is we will apply a safety factor. We can't check it on human beings. Just like we took 10 rabbits, we can't take 10 human beings and we can provide them with 10 different dose of pesticide with daily in their diet. We can't do it. It's not ethical. So what we will do is we will apply a safety factor. We have no observed adverse effect level, and we will divide it by 100. That is a mammalian safety factor. We will divide it by 100. So Abhishek has told us earlier, told 1 milligram per kg body weight per day is safe for rabbits. So what he will tell is divide it by 100. So that will be 0.01 divided by 100, and he will say that 0.01 milligram per kg body weight per day, if human beings consume, it will not cause any adverse effect to human beings because I applied the mammalian safety factor to it. And that value NOAEL divided by the mammalian safety factor, that is 100, is called as acceptable daily intake. Are you clear now what is acceptable daily intake? I'll come into the definition later. Are you clear the concept of acceptable daily intake? Yes. So I will give the definition. It's the maximum concentration of residue. Again, it is the maximum concentration which, when ingested over the entire life period, will not cause any adverse effect based on the knowledge at that time. Again, it is milligram per kg body weight per day. I have forgotten to add per day. I will add it now itself, otherwise forget it again. So it's the same maximum. Only difference is that no observed adverse effect level is in test animals. Acceptable daily intake is with respect to human beings. So it's the maximum concentration of residue which, when ingested over entire lifetime, will not cause any adverse effect. One thing, the next thing is critical, based on the knowledge at that time, because we have studied up to that, because certain pesticides, its adverse impacts get manifested after a prolonged usage only. So we may not say, can say that we have checked this, it is safe and it will be safe for the entire time, for say 10 years, 20 years, 30 years. No, we can't say that. Because after it's released into the market, we'll get data from different people, and that data may point out to some sort of adverse effect. So in that case, we should go for a review. So that is based on the knowledge at that time. So that is one term which is added to it. So it is nothing but the maximum concentration of a residue which is ingested, which when ingested over entire lifetime will not cause any adverse effect based on the knowledge at that time. Okay, this is acceptable daily intake. This okay, this is acceptable daily intake. Then comes maximum residue limit. Maximum residue limit is the definition says it is the maximum concentration of a residue that is legally permitted or recognized as acceptable. Maximum concentration of a residue that is legally permitted or recognized as acceptable in food, agriculture, commodity, or animal feedstuff when the pesticide is used under supervision, adopting GAP, good agricultural practices. I will explain. That is the maximum concentration of a pesticide that is legally permitted or recognized. It is a legal value or recognized as acceptable in food, feed, or animal agriculture commodity when the pesticide is used under supervision, adopting good agricultural practices. I will explain it. Say a farmer has come to you. He has having some problems. Say bitter gourd, fruit fly in bitter gourd. You have identified that. Rahul has identified it as bitter gourd fruit fly, and he has recommended a particular pesticide. And he recommended that pesticide, and he recommended it at the rate of 2 ml per liter. And he advised the farmer that there is a waiting period of 3 days, so don't harvest the crop within 3 days, or you can have to, once you spray the crop you should harvest only after third day, and the dose is 2 ml per liter. Okay, but the farmer goes to the field, instead of 2 ml per liter he takes 3 ml per liter, and he harvests the crop on second day. So what will be the thing? Whether it constitute good agriculture practice? No, no. So that is the thing. We can only say, see, with respect to the pesticide residues, we can only say that, say, if you are using a particular pesticide, and the dose is, say, 2 ml per liter or 3 ml per 10 liter, if a farmer is using a particular pesticide in a particular crop, and if you are harvesting after this much waiting period, the residue will be this much. Because if harvesting the next day, second day, third day, fourth day, residue will vary. If the farmer changes the concentration of the pesticide applied, again the residue will vary. You are only sure about that particular dose, and after this much period what will be the residue. So that is MRL, maximum residue limit. It says that the maximum concentration of a pesticide residue that is legally permitted or recognized as acceptable in feed, food, or agriculture commodity when the pesticide is used under supervision following good agriculture practices. So if a farmer doesn't follow good agriculture practices, the residue will be varying. So we usually give MRL value based on good agriculture practices. We usually say that if you use this much concentration of pesticide, and if you follow the prescribed waiting period, the maximum concentration of pesticide residue which is present in a particular crop is this much, and we will notify it in Gazette, that is legally acceptable. And this is purely a trade limit, it is not a toxicological limit. That I will explain. This concept you have to understand. This is purely a trade limit, and this does not mean that it is having some toxicological significance. That means you have consumed some apple today. Shiga purchased some apples. She consumed it, and after consuming the 2 or 3 apples, she felt something bad in that apple. She felt that something is not correct with that apple, and she brought that apple to the lab, and we tested it over here, and we found out some pesticide residue, and that too above MRL. So what will be the reaction of sugar? What do you feel after you have consumed three apple? Whatever the residue analysis results will be ready after. If you do it quickly, it will take at least 3 to 4 days. So already you have consumed three apples. You felt that something is there, and we checked it. Yes, there was some pesticide residue, and that residue is above MRL. So what will be your feeling, Shika? What will you derive out of this results? Fear. So what fear will you have?
I have
Will you get?
consumed some toxic,
Huh?
I have consumed some toxic substance.
Consumed some toxic substance and
Toxic
it can
substance. So
cause some adverse health
I
effect to
wonder
you.
what
Okay,
about
okay, okay. So keep it that in mind. I will ask the question later again. So how we calculate maximum residue limit in our country? That also I will explain. So you might have heard about All India Network project on pesticide residues. It is in Kerala. That center is in Vellaini. Have you heard? Yes. So we have such about 20 or something centers all over our country. So if a particular company is introducing a new molecule, so what they will do is for residue estimation, for MRL estimation and all, they will give that sample of pesticide to all these centers, different centers. Take 20 centers, different centers, and that they want to check it in rice. Okay, so all the centers cultivate rice, give spray the pesticide as per the protocol given by the company. They will say that spray the pesticide when leafholder population is this much, or when the crop is at the maximum tillering stage. So at the maximum tillering stage, all the stations sprayed the particular pesticide, and they harvested the produce and they checked the residues in the grain. And these 20 locations will report different value. Kerala will have high rainfall. Rajasthan, Gujarat will have high temperature, and Himachal Pradesh and all will have low temperature. So dissipation of pesticide will be different. In Kerala, in Vellaini people, when once they sprayed the pesticide, after 3 hours they had a heavy shower. Kerala, we can expect that. So there will be a lower pesticide residue in Kerala. In Himachal, it is extremely low, so temperature is extremely low, so residue can remain longer. So they will report a higher residue value. So 20 locations, if you are checking residue value from 20 locations, even though they have sprayed the same pesticide at the same time at the same concentration, the residue detected can vary. That's natural. So what we will take is that among the 20 centers, which center reported the maximum concentration of residue? We won't take the average of 20 locations. We won't take. Say Himachal Pradesh center reported the maximum residue. They reported that if they spray that particular pesticide at that in a particular stage of the crop, and if we harvest the produce, we will get this much pesticide. They will say that they will get 0.01 ppm of pesticide residue. All other centers reported that at the harvest we didn't had any pesticide residue at all, and only Himachal Pradesh center reported that we had pesticide residue of 0.01 ppm. Then we won't say that among the 20 centers, 19 centers reported any doesn't reported that there is no residue. Only one center reported 0.01 ppm. We can't take other values. We will take only the highest value. We will take 0.01 ppm of Himachal Pradesh. So that is maximum residue limit. That is the maximum possible residue, because MRL is for the whole country. FSSA declaration, FSSA gazette notification is for the entire country. So that means if any farmer in any part of the country spray that particular pesticide, this is the maximum at the particular concentration at the particular crop stage. This is the maximum concentration of pesticide residue that we can expect. Clear with respect to MRL? Yes. Okay. Then comes earlier Abhishek was discussing something about the weight will be different. Then I told it's different. We will take the weight later. That is one thing. Maximum permissible intake is one value wherein we take out the body weight of the person. We acceptable daily intake is for milligram per kg body weight per day. Maximum permissible intake what we will take is for human beings. There is average body weight for Indian Indian Council of Medical Research, ICMR, gives the average body weight for Indians. They have average body weight for men, Indian men, average body weight for Indian women. So they have a statistics for that. So what we will do is for Indians, for a particular pesticide, they will what they will do, we have the ADA value and we will multiply it with the average body weight of a Indian, say 65 kg. So the ADA as calculated by Abhishek in the earlier example was 1 ppm, and for Indians he will work not ADA, NOAEL was 1 ppm. Our ADA, after dividing the mammalian safety factor by 100, it became 0.01 ppm. So that is per kg body weight. For Indians, if the value is the average body weight is 65, then the maximum permissible intake is 0.01 into 65. Are you clear what is maximum permissible intake? Yes, that will become 0.65. So what is your body weight, Abhishek? Rahul and Shiga, you can just type your body weight. Rough, you might have gained. Rahul is 69, Shiga is 45, Abhishek is 69. So Rahul is the person who can consume the maximum quantity of pesticide. Shiga is the person who can consume the least quantity of pesticide, because you have the least body weight, so you can consume only less quantity of pesticide. So that's the thing. But we don't take individual values, we have to give a national value, so we take average body weight. So that is maximum permissible intake. The maximum permissible intake will be lower for Shiga and higher for Rahul and Abhishek. Okay, so that's maximum permissible intake. I don't know whether I should take, it will take some time. Okay, quickly I will complete it, because if I do it tomorrow, it will take more time. Okay. Uh, Shiga was telling that she has consumed an apple and it had residues above MRL, and she has feared that she will get some adverse health impact because of consumption of pesticide. We will check it out how we calculate the dietary risk assessment. We have uh, told one term, dietary risk assessment, that is theoretical maximum daily intake. So I'm stop sharing and I will be sharing my entire screen, and who will be willing to share your yesterday's diet? He or she can unmute and tell. നിങ്ങൾ ആരെങ്കിലും നിങ്ങൾ ഇന്നലെ കഴിച്ച സാധനങ്ങൾ ഒന്ന് പറയണം, എന്നിട്ട് വേണം നമുക്ക്. നിങ്ങൾക്ക് ഇപ്പൊ ഒരു എക്സൽ ഷീറ്റ് കാണാവോ? അൺമ്യൂട്ട് ചെയ്തോ, നിങ്ങൾ അൺമ്യൂട്ട് ചെയ്തോ, 3 പേര് അൺമ്യൂട്ട് ചെയ്തോ.
Sure.
Are you able to view an Excel sheet now?
Yes, sir.
Yes. Okay. Who is going to share your yesterday's diet? ആരാ പറയാൻ പോകുന്നത്? രാഹുലെ പറഞ്ഞോ. So what you took immediately after getting up today or today or yesterday? രാഹുൽ ഇന്നലെ കാലത്ത് ആദ്യം കഴിച്ചത് എന്താന്ന് ടൈപ്പ് ചെയ്യരുത്. ഞാൻ ടൈപ്പ് ചെയ്ത് ഞാൻ അതിലേക്ക് പോയാൽ അതുകൊണ്ടാണ് ഞാൻ അൺമ്യൂട്ട് ചെയ്ത് പറയാൻ പറഞ്ഞത്. രാഹുൽ ഇല്ലേ? രാഹുല് ലീവ് ചെയ്തല്ലോ. ഭക്ഷണം ചോദിച്ചപ്പോ രാഹുല് ലീവ് ചെയ്തു. ശിഖ പറഞ്ഞോ.
ദോശയും ചമ്മന്തിയും.
ദോശയും ചമ്മന്തിയും. Okay. So what are the constituents of dosha? Rice.
Rice and black gram.
Black gram. Okay. Then chutney. What all things you add? Coconut.
Coconut. Oil.
Oil. Mustard.
Onion.
Mustard, chili.
Curry leaf. Chili, curry leaf.
Chili, curry leaf. Okay. So, how much quantity of rice you must have taken in your how much dosha you would consume? 2, 3, 4? 3. Okay. So what roughly approximately can you say how much quantity of rice you have taken?
Lunchino.
അതായത് ഈ 3 ദോശയിലും കൂടി എത്ര അരി ഉണ്ടായി കാണുന്നു? What is the quantity of rice that constitute that must have gone into preparing this 3 dosha? Roughly, I will have 50 gram. Okay.
Okay.
50, is it? No, I think it will be smaller. I will take 100. Okay. What is the proportion of rice and black gram in dosha? 1:1?
2:1. 2:1. Rice is 2.
2:1. So then it will be 50. Okay. So for every 100 gram, you might have taken 50 gram of black gram. Okay. For chutney, how much grams of coconut you might have taken?
Maybe 25 gram.
Okay, 25. Oil.
Oil.
5 gram. I will take 5. Mustard 1 gram. Onion 5 gram. Chili 5 gram. Curry leaf 1 gram. Okay. So what did you take tea in the morning?
Black tea.
Black tea. So I will add tea powder. It will be roughly 10 gram. Okay. So that was for finished your morning. Then what was there for lunch?
Rice.
Rice. How much quantity? 300 gram or 400 gram?
300 gram.
300 gram. Okay. Then
more.
what are the for curries?
Morari and Goni.
Morari. Okay. So what again there will be coconut in that, I feel. What all the ingredients you can tell? Coconut you can take it as 10. Then oil will be there anyway.
Oil will be there.
One. Mustard will be there again 1. Curry leaf will be there 1. Then chili will be there 1. Is it turmeric?
Yes.
Again 1. Then coriander?
No.
Huh?
Curry leaf. No,
No.
coriander plant.
Oh, okay.
Curry
Then
leaf.
banana,
Oh, it's OP melon.
OP melon. Okay. OP melon. That can be 25 gram. Okay. Okay. Then what is there for others?
Um, and then omelette.
Omelette. Egg. I will avoid egg because we don't use pesticides on poultry, so I will avoid egg. Anyway, you might have used onion.
Uh, oh yes.
I will add onion over here. Again 5 gram. Oil again 1 ml. Then
Chili.
chili 1. Okay.
Mm,
Okay. Then omelette. Any other thing?
cabbage. Cabbage.
Cabbage.
Carrot toren. Mixed toren.
Cabbage. Okay. I will add cabbage. It will be something 50 gram. Again oil. Mustard will be there. 1. Chili will be there. 1. I'm roughly calculating. Okay. So the night also you might have taken what night what you have taken quickly you can tell. I'll just calculate it.
Chapati.
Chapati. So there will be wheat. It will be about 200 gram or 250 gram.
200 gram. Okay.
Okay, 200. Then what else?
Mm, chicken curry.
Chicken. Okay, meat. Again, it will be 200 gram. Again, there will be onion.
Oil.
The onion will be there a bit more, 20. Chili will be there, 2. Curry leaf will be there, 1. Oil will be there, 1. Okay. So, did you get how we calculate it? Every day, whatever you consume, you have to calculate. I am summing it up, all the things. Okay. So I'll add one more thing. You took apple. Consider you took apple. 3 apples you took. So that will be roughly 200 gram. Okay. Okay. Fine. So we consider a typical example of pesticide as quinalphos, Ecalix. So there will be MRL for quinalphos in rice, MRL for quinalphos in black gram, coconut. Every commodity the MRL will be different. Oil will have MRL, mustard there will be MRL. Different commodity we have MRL for quinalphos. So MRL the unit is milligram per kg. So if we usually consider that the MRL, whatever be the commodity which we consume, it is having the residue at MRL level because we theoretically we say that all the commodities will have the residues at MRL level, even if it contains pesticide or contains pesticide or not. Sometimes the commodity may not have pesticide, some the commodity will have pesticide. Okay, so in the rice Kinalfo MRL is say 0.01 milligram per kg, 0.01. I will give the same value for all. MRL is same for all the commodities. It's not so. It will be different, but take it will be the same milligram per kg, and we will multiply. Sorry, this into this MRL because we haven't point no. we usually consider food factor. We won't be taking 4 milligram. It will be oh sorry. This is zero point 0.4. We are we took grams. This is gram. We are calculating it in milligram per kg. So we have to convert it into kg. So that means into 10 raised to minus 3. So that's why. Understood. This should be in should have been in kg. It is in gram. So that's why is equal to 400 into this. Okay, this is the quantity of quinol force Shikha has consumed yesterday through different diet. I will sum it up. Okay, Shikha has consumed 0.01226 ppm of quinol force yesterday all through the diet, and from the entire diet she has consumed yesterday. So this is with respect to the entire diet. What we are imagining that the entire commodity, all the commodity, rice is having quinol force, black gram is having quinol force, coconut oil, mustard, everything is having quinol force residue, and adding it up, we theoretically we say that Shikha has consumed 0.01226 ppm of quinol force yesterday, and we will compare this value with the ADI value, acceptable daily intake value for quinol force. See, the ADI for quinol force is I'll just check for ADI for quinol force. Quinol force it is 1.91 microgram per liter. You can see that value 1.91 microgram per liter. Microgram per liter is ppb. So it will be point 00191. That is the acceptable daily intake for quinol force, and she has consumed 0.01. So in that case, we will say that the quantity consumed is bit more, and it can have some deleterious effect if she consumed this much quantity daily over his entire lifetime. Again, you can think that this is daily. That means if she consumes apple daily, if apple is removed, uh, if apple is removed, if it doesn't consume apple tomorrow, then this value will be decreasing. If so, if this thing is subjective, and every day if she is consuming all those things, and every day all the commodities are having the quinal force, then only we can say that we have this pesticide residue above. We compare with ADA with the MRL value, then we will say that we have pesticide residue which is somewhat harmful to us. So this is the way we calculate dietary risk assessment. Are you clear regarding this? Otherwise, I'll repeat it again tomorrow. Hmm?
ഇതെങ്ങനെ ബോഡി വെച്ച് കമ്പയർ ചെയ്യാൻ പറ്റുന്നത്?
എ ഡി ഐ വന്നപ്പോ ഇവിടെ ഒരു കാര്യമാണ്. ഇവിടെ സോറി, ഇവിടെ എ ഡി ഐ വെച്ചല്ല, ഇവിടെ തിയറട്ടിക്കൽ മാക്സിമം ഡെയിലി ഇൻടേക്കും കൂടെ വന്നില്ല. മാക്സിമം പെർമിസിബിൾ ഇൻടേക്കുമായിട്ടാണ് ആയിരുന്നു കമ്പയർ ചെയ്യേണ്ടത്. സോറി. സോ ഇറ്റ് വിൽ ബി അഗൈൻ ഇൻടു 65. ADA is point. Okay, I will share the screen again. Sorry, that was a relevant thing. That was a relevant thing. I forgot to do that. It should not have been with ADA. It should have been with the maximum permissible intake. So ADA is this much. Our average body weight is average. Okay, Shika's body weight I will take. Body weight 45 is equal to into. So now you see. So ഇപ്പൊ ശിഖ എന്ത് പറയുന്നു? Do you fear? What will be your reaction now after this?
There's nothing to fear.
അങ്ങനെ
പേടിക്കേണ്ട ആവശ്യമൊന്നുമില്ല.
പേടിക്കേണ്ട ആവശ്യമില്ല. So this is the way we calculate. How? Are you clear, Rahul and Abhishek? ക്ലിയർ ആയോ എങ്ങനെയാണ് നമ്മൾ ഒരു ഡയറ്ററി റിസ്ക് അസസ്മെന്റ് വർക്ക്ഔട്ട് ചെയ്യുന്നതെന്ന്? How we work out dietary risk assessment based on our diet? സംശയം ഉണ്ടെങ്കിൽ നിങ്ങൾ അൺമ്യൂട്ട് ചെയ്ത് ചോദിച്ചോ. You can unmute your mic and ask. Clear. Rahul? രാഹുലിന്റെ നെറ്റ്വർക്കിന് എന്തോ പ്രശ്നമുണ്ടെന്ന്, ഓഡിയോ വരുന്നില്ല. ഓക്കെ, is it clear? Yes. But one thing you should keep in mind is that this is a theoretical scenario. Usually, we represented wheat as having quinal force, but wheat is one crop where we normally doesn't use pesticides at all. Usually, the only pesticide used is weedicide, not insecticides, and sometimes fungicides are used, but not insecticides. So we won't get any residue from wheat. Then curry leaf, that's one culprit. Chili we may get, but oil, the chances of residues are relatively low. Onion, the chances of insecticides are very less. So even though we have said that these commodities are having pesticides at MRL, practically we may not get residues in at least half of the commodity. So that, and we must consume this quantity every day. That the quantity, if the quantity is above acceptable, the theoretical maximum permissibility MPI every day, then only we are at risk. Otherwise we are not at risk. So I think tomorrow I will just ask your friends to go through the recorded video. I will upload this recorded video today itself in the Moodle. Ask your friends to go through it. If they feel any doubt, we will discuss it tomorrow and we will proceed for this. One more concept we have to tell that will take another 10-15 minutes. So I'm not going into that. Are you clear regarding this concepts? Yes or no? Because you have to be extremely careful, because some persons spread the fear that if the value is above MRL, that commodity is unfit for consumption. Do you have that opinion now? If you check for any pesticide residues in a particular commodity, and if it is above MRL, it doesn't mean that it is unfit for consumption. It's not so. That's why I told you one sentence in the case of MRL, that is, it is a purely a trade limit and it is not a toxicological limit. If the residue crosses a MRL, it doesn't mean that it will cause some health effects on you. It's not a toxicological limit at all. It's a trade limit at all. I will give one example and I will finish off the class. Have you heard about a fungicide called as tricyclosol? Its trade name is Beam. Yes, it is used in rice for seed treatment in rice, especially in basmati rice and all. So, what the European Union fixed the MRL for tricyclosol in basmati rices 0.01 ppm, and US EPA, the American Environmental Protection Agency, fixed the MRL for tricyclosol in the same basmati as 1 ppm. I will repeat. European Union fixed the MRL for tricyclosol in basmati rice as 0.01 ppm, and US EPA fixed the MRL for tricyclosol in again the same basmati rice as 1 ppm. Basmati rice is produced only from India and Punjab, and it is consumed all over the world. It is exported heavily. So this doesn't mean that Americans have greater tolerance for tricyclosol and Europeans have lower tolerance for tricyclosol. It's just a trade limit. If European Union doesn't want the import of basmati rice into their country, what they will do is they will keep the MRL at lowest level possible. Because that I will discuss it in tomorrow's class, how companies are fixing MRL and all, because they are not growing basmati rice in their country, so they can fix whatever value they wish. If India is fixing MRL for one particular, say, basmati rice, if you are cultivating basmati rice in our country, the MRL value should be based on our trial, our data generated in our country. But if you are not cultivating a particular crop in our country, we can fix value according to our wish. So that's what EU and US has done. They can't cultivate basmati rice in their country, so they fixed an arbitrary value, and that arbitrary value is mainly based on a trade decision. If they want to decrease trade from a particular import from a particular country, they will fix it as low. I will give one more example and we'll finish up the class. Saudi Arabia, they cultivate only one commodity, date palm, so they can fix MRL only for one commodity. You learned how MRL is fixed. That means they have to cultivate the crop, they have to spray on that particular crop and generate the MRL data. But they have desert and they are growing only date palm, and they are importing all other food commodities from other countries. And what they did was they fixed for all commodities, food commodities, as 0.01 ppm. That is 0.01 ppm is the one which can be detected in instruments now, which can be the lowest level which can be detected in instruments is now 0.01. So they kept the value as 0.01. So this is the way countries try to reduce the trade balance and trade imbalance, reduce the import and all. So this is purely a trade limit. This is not a toxicology. Understand the, yes, as Abhishek Gowda said, it is marketing strategies. If Americans doesn't want mangoes from our country, they will keep the MRS at a pretty low level. If India, the problem is we have diverse climatic condition. We can grow almost all the crops, so we can't arbitrarily fix a MRL value for a particular pesticide, because we have most of the crops grown here. If we have crops grown in our country, we have to generate MRL data from our country itself. Okay, I'm stopping it. I've gone a bit longer because this is a thing which can't be stopped in between. So